Today I will be discussing first Nirenberg and Matthaei's experiment and second Nirenberg and Letter's experiment, both of which contributed greatly to cracking the genetic code. The question that Nirenberg and Matthaei's work centered around was about nucleotides made of one base and which amino acid they corresponded to. So the codons of interest were UUU, AAA, CCC, and GGG. In order to discover which amino acid went with each of these codons, Nirenberg and Matthaei developed an experimental design in which a long chain of uracil nucleotides was combined with a cell-free extract in all 20 amino acids. They marked one of these amino acids as radioactive. They then allowed the uracil polymer to translate, and then they filtered the solution to see if the translated protein had become radioactive. The idea was if it had become radioactive, then the UUU codon coded for the amino acid that was chosen to be radioactive at the beginning of the experiment. This method allowed Nirenberg and Matthaei to discover that UUU codes for phenylalanine, AAA codes for lysine, CCC codes for proline, and GGG did not code for an amino acid. This experiment was the start to cracking the genetic code. So Nirenberg and Letter picked up where the last experiment left off to continue cracking the code. This time the question was, what if the codon isn't composed of only one base? What amino acids do all the other codons code for? To figure this out, Nirenberg and Letter combined one codon, a ribosome, and 20 charged tRNAs, each with a different amino acid. One of these tRNAs was made radioactive. The codon bound to one of these tRNAs. The solution was then filtered out. If it was not radioactive, the procedure was repeated with a different amino acid marked as radioactive. If the filter paper was radioactive, it was known which amino acid the codon coded for. Through this procedure, Nirenberg and Letter discovered 61 more codons, each of which code for a specific amino acid. Combined with the 3 that were discovered from Nirenberg and Matthaei's work, this gives us all 64 codons that make up the genetic code we know today. Through their respective experiments, Nirenberg, Matthaei, and Letter all contributed to successfully cracking the genetic code.